Alright, so as Amanda said, today I'm going to be talking about the gut-brain connection and how the health of your gut might affect your mental health. The gut-brain connection is actually a real connection. There's a nerve called the vagus nerve that connects our gut and our brain, and the role of it isn't fully known yet, but this paper is going to partially illuminate one of its roles. So my paper is called Regulation of Prefrontal Cortex Myelination by the Microbiota, or in more simple terms, exploring the connection between the gut microbiota and neuropsychiatric diseases. So before I get started, some important terms that are going to pop up a lot throughout the presentation. These are going to be kind of important to know, and if you have any questions during the presentation, just ask me about them. First is myelination, which is the process in which nerve fibers are insulated on axons, and it's essential for all axons. So as you can see in this picture here, the kind of darker black line, that's the myelination. It's a fatty substance that's put in there by oligodendrocytes in our brains. Next is axons, obviously, and those are the thread-like part of nerve cells that conduct impulses from one cell body to another. And you can see an axon right there. It's the thread-like part of it, and the yellow on this is actually myelin sheaths. And as you can see, they're all connected to each other, so impulses are able to be conducted properly. Next is the gut microbiota, which are all the trillions of microbes that live in our gut. There's at least 1,000 different types of bacteria in our gut, and all of them are essential in order to fulfill different functions that we as humans can't fulfill. So for example, they might provide nutrients that humans can't make for themselves. They fight off foreign bacteria that we don't want in our bodies, and then they also digest food. So we couldn't live without our gut microbiota. A few more. So the prefrontal cortex is what regulates social, cognitive, and emotional functioning in our brains. And it's like the frontmost part of the brain. Does anybody here know what upregulation is? Which is my next term. No? Okay.
Yeah, it's like the introduced expression of something.
Yeah, so it's the increase of a cellular component that's in response to an external variable. So in this paper, RNA will be upregulated or downregulated a lot, and that'll be kind of important. So then downregulation is the opposite. It's a decrease of the cellular component. And then lastly, demyelination, which is the loss, damage, or downregulation of myelin, and it's what causes or is related to many neuropsychiatric diseases. So as you can see in this is a regular axon. There's like a full, thick myelin sheet, and all of the connections can be properly made because they're not damaged. But in this one, which I think is an axon with multiple sclerosis, there's damaged myelin sheaths, as you can kind of see here, and the connections can't be made. And that's what causes this neuropsychiatric disease, which brings me to my next slide, all of the neuropsychiatric diseases that are related to demyelination. So things that we hear about every day, like autism, schizophrenia, depression, anxiety, Parkinson's, sclerosis, all of these diseases are related to demyelination in the brain. And like I said before, in a normal axon, where there's fully insulated myelin sheaths that are all connected, impulses are able to be conducted properly. But in the multiple sclerosis axon, there's really damaged myelin. The connections can't be made. There's too little myelin, essentially, and that's what causes this neuropsychiatric disease. So in my paper, they looked at 3 different groups of mice, and they did a genome-wide transcriptome profiling approach, which means that they extract all of the RNA transcripts from the tissue of different parts of the brain in these mice, and they examine them, they analyze them, compare and contrast the different areas in the different parts of brain between the different 3 groups. And things that they might look at are things such as upregulation or downregulation of the RNA and different types of RNA. So the first group of mice that they looked at were germ-free mice, which are mice that are bred in a lab and kept in an incubator, because germ-free mice don't have any germs in their stomach, which obviously, as I said before, no organisms can live without this gut microbiota in their stomach because we need it to survive. So they're kept in an incubator, so there's no germs that are allowed to go into that incubator. The food is very special that they get without any germs on it, so they can be kept germ-free. The next group of mice were ex-germ-free mice, which for the first three weeks of their life, they were germ-free, but after 3 weeks they had germs introduced into them. And the effects were really interesting from that. And then the last group were conventional everyday mice that just like you and I have germs in our stomach that we need to survive. And so what they did in the paper is, as I said before, the ex-germ-free mice that were originally germ-free and then had germs introduced them, they had the germs introduced after 3 weeks. All of these tests with the genome-wide transcriptome profiling approach was done after 10 weeks. So it's gonna be important later in the paper, the 10 weeks thing. So the first thing that they looked at was the location of upregulation of many of the genes and where it did occur and where it didn't occur. So the green bar in here is the germ-free group. And as you can see in the prefrontal cortex, which is what this graph is showing, there was upregulation of myelination only in the prefrontal cortex. So all of these genes, MAG, MVP, are all related to myelination, and all of the RNA transcripts are upregulated in the prefrontal cortex, whereas the black and blue bars, which are the conventional and ex-germ-free groups, they all kind of have a normal level pretty much consistently throughout. So if we were to look at the other parts of the brain, like the cerebellum or the amygdala, this green bar wouldn't be irregular like this and really high. It would just be the same across all three groups. So this only occurred in the prefrontal cortex. The next thing they looked at were the different genes that were upregulated and what their function were in the germ-free mice. And what they determined is that certain genes like EGR2 in the germ-free mice, which is again the green bars, have a significantly higher amount of transcription for myelination upregulation factors compared to other genes in the germ-free mice. Some of the things that the EGR2 gene does is it has to do with oligodendrocytes, which are the type of cells that produce the actual myelin sheath, or like the fatty substance that connects the neurons, and or just any myelin-specific genes. And then not all genes, so in this graph, SOX2 and SOX10, coded for myelination were like significantly upregulated. What the researchers found is that almost 15% of all of the upregulated genes in germ-free mice in the prefrontal cortex had to do with myelination. So what we can, like just hearing this 15% thing, you can assume that the myelin sheath around a germ-free mouse, around the axons of a germ-free mouse, is going to be really, really thick, which is a good thing because, like I said before about the neuropsychiatric diseases, you kind of want that extra buffer of myelin in order to have all the axons remain connected and properly insulated. So, as you can see in these images right here, when they examined images of axons extracted from the prefrontal cortex of both germ-free and conventional mice, you can see in the germ-free mouse there's a really thick myelin sheath. And then the conventional mice, there's just like a normal myelin sheath. It's not damaged or too little as of right now, but it's just normal. Okay, so just a question to make sure everybody's understanding so far. If conventional mice have a smaller myelin sheath, or just like a normal myelin sheath, what might this mean in the context of demyelinating neuropsychiatric diseases? Like, if they have a smaller myelin sheath, would they be more prone to a neuropsychiatric disease or less prone? Katie? Exactly. So, because they have either this damaged myelin sheath, because they have just a normal myelin sheath, they have a higher susceptibility of getting a neuropsychiatric disease like depression, anxiety, schizophrenia later in their life, or they might have a higher chance because they don't have that extra myelin sheath as a buffer. And the opposite question, if germ-free mice have a thicker myelin sheath, what might this mean in the context of demyelinating neuropsychiatric diseases?
They're less likely to get disease later in life.
Exactly. So this would be good. this is a good thing for these germ-free mice. They have this extra myelin sheath, and that means that they're less prone to getting these neuropsychiatric diseases. So the next thing, this is kind of hard to understand, so stop me if anyone has questions. The next thing that they looked at was the G ratio of axons in the germ-free and the conventional group. So what G ratio is it's the ratio between the inner axon diameter to the outer axon diameter, which includes the myelin sheath. And it's an inverse relationship, so a higher G ratio would mean there's less myelin, a lower G ratio would mean there's more myelin. So as you can see in figure D here, the diameters for the inner axons are pretty much the same. They're very close, the bars in figure D, for the conventional and the germ-free group. But the germ-free had, like we said before, the germ-free had more myelin, which means a higher outer diameter and a lower G ratio. And a lower G ratio means more myelin, whereas the conventional mice had a lower diameter because they just have that normal amount of myelin, so it's a lower diameter, which means that they had a higher G ratio and less myelin in the conventional group. Something else that was really, really interesting that the scientists also measured was the protein concentration in the three groups. Conventional, which is the group that is born with the germs, germ-free, which is the group that was born without germs, and then X-germ-free, which is the group in blue here, that was born without germs and then at 3 weeks had the germs reintroduced to them. And what they found was that the X-germ-free mice still have this increase in myelination and proteins related to myelination at the 10-week mark, which is when they measured this. So what this means in the context of humans, if we're gonna bring this into the big picture of the paper, would we be able to replicate this in humans? The X-germ-free mice still had this positive effect of extra myelination, which is something that we would potentially want, but they still had germs introduce them after 3 weeks, and they still got this positive effect. So what this kind of means is that you don't have to be germ-free forever in order to get this positive effect that we saw in the germ-free group, which still had this increase in proteins. The X-germ-free group still got this positive effect and just didn't have the germs for the first 3 weeks of their life. So what this might mean is that in order to get the positive effect of an increase in myelination, it has to be during one of the critical periods of development in the brain, which is either when you're first born, so like the first 3 weeks of its life, or during adolescence. These are when most of these positive changes might occur. So in this paper, they determined the effects of the total absence or the total presence of the gut microbiota. And what they determined is that if the microbiota in these mice was absent, then there would be an upregulation of myelination in the brain. And if it was present, then there would just be a normal amount of myelination in the brain. And then also from the slide before, they determined that being X-germ-free for life or permanently isn't necessary because these X-germ-free mice still had this same amount of increased myelin proteins, which is something good that you'd want. This paper is interesting because it kind of opens up the door to a lot of more future research. So in this paper, they determined the total absence of the gut microbiota, but they didn't determine if, let's say we were to take out part of the gut microbiota, if we'd have similar or the same effects. So as I kind of told you guys in the beginning, we can't live without our gut microbiota. We need it in order to survive. So if we were to try to replicate this in humans, then it wouldn't be realistic to just take out all of our gut microbiota because we wouldn't be able to function. So what scientists in a future study need to do is they need to determine if there's specific strains of specific strains of bacteria that are causing this effect. So, for example, it might just be 1 or 2 strains of bacteria that cause an altered level of myelination. In that case, we wouldn't want to remove the whole entire gut microbiota. We might just want to remove those 1 or 2 strains of bacteria. And one way that scientists are able to determine this is called monocolonization, which allows us to see how just one strain of bacteria might affect the altered levels of myelination, because what they do is they'd introduce, this would take a really long time, but they'd introduce just one type of bacteria into the gut of a mouse and then see how the myelination is affected or normalized by that one bacteria. And they can do a few at a time too to see how that aggregate of bacteria affects it. And then also, this was mentioned really, really early in the presentation, but the vagus nerve, which is the nerve that actually connects our gut and our brain, we don't know the exact role of it yet. We know some of its roles, and the vagus nerve, again, it connects the gut and the central nervous system, which is the brain, the spinal cord, and the sensory system. so we don't know if the vagus nerve could also lead to altered myelination levels. And then also like making it even more abstract. So we don't know whether this is a preventative, if it's just a preventative for demyelinating disease possibly, or whether it could be a cure. So what that means is that like someone who already has, for example, sclerosis, can we take away certain germs in their stomach and see if that would cure the disease? We don't know if that would work, or whether it has to be in younger kids. We prevent them the disease before they happen. So again, this is a possible pathway to treating the actual illness instead of just treating a symptom. So a lot of times, for example, with depression, which is one of the demyelinating diseases, for example, mood might be treated, which can be effective, but it doesn't always work for all people, and it doesn't always work forever. It might only work for a period of time. So this would be actually treating the root of the disease rather than a symptom of the disease. And then also scientists need to determine when is the ideal time to take these bacteria out again. For example, adolescence, or when you're first born, or two of them. And then finally, there's some new treatments that open up based on this, that are based on prebiotics, probiotics, and fecal transplants. And what those basically are is when you extract the feces from a human, and then you reintroduce a new feces into that human that has only specific bacteria strains that we really want, or that we want to see the effect of them. Thank you. Does anybody have any questions?
So do they know if demyelination is the sole cause of these neuropsychiatric diseases, or is it a contributing cause?
Well, it depends on the diseases. So I know for multiple sclerosis that is one of the main causes, but for some of them, there's more than one cause.
So does the amount of myelin, right, that you have, does it deteriorate over time on its own, or is it kind of directly related to the risk?
So when you're born, for the first 30 years of a human life, the myelination levels in your brain are changing. So after that, it kind of stays pretty constant, and then from there, it might get damaged, or there might be a loss of myelin from there. So it's not ever changing, and that's why certain neuropsychiatric diseases are more prone to people later in life. Any other questions? Jeremy?
You mentioned that having no germs could harm us and the mice. For the first 3 weeks of the germ-free mice, did any mice get hurt or die?
That wasn't mentioned in the paper, but they lived in a really controlled environment because, in order to be germ-free and not become a walking bacteria trap, they had to be kept in a special incubator with all the other germ-free mice, and they had to be given special food from the scientists that had essentially no microbiota on it, so they wouldn't become infected with bacteria. So I think that kind of answers your question. I don't think any of them died because they're in such a controlled space. Any other questions?